Yogah Karmasu Kaushalam.
Namaste. This is Dr. Sindhav Gaurang, Assistant Professor at Gujarat University, University School of Sciences, Department of Zoology, Biomedical Technology, Human Genetics, Wildlife Biology and Conservation. Being a family member of Gujarat's parents as well as an esteemed and the biggest university, we welcome you all to this virtual second online refresher course in life sciences organized by UGC HRDC Gujarat University. Here, I would also like to convey wholehearted thanks to the team HRDC and Gujarat University for giving me the opportunity to connect, interact, and share my views with all you wonderful friends. Hope you all are safe and sound. As we all know, this time it is quite different and like survival of the fittest. પરિવર્તન અને નવરચના એક સિક્કાની બે બાજુ છે જેમ આપણે ગુજરાતીમાં સરસ વાત કરી છે ને innovation and adaptations leads us toward e-learning and the show must go on. So please, I request you all to join me to inaugurate my today's interaction with a little warm-up. Sometime, somewhere, we all have experienced a very basic question, and of course, the important question in our life, or rather say, most of the time when we are weak or in some dilemma. The glass philosophy. Is the glass half empty or half full? We have been called, we have called as optimist or pessimist. Depend on observations, as many motivational speakers inspire us that we have to be optimist. And yes, of course, why not? But as an inner scientist, if we see it is full of sciences, if we think beyond the bounds, it is always full. How? Let's take a look. As a thinkist, hope I'm making it clear. We can see always glass full. Yes, it is always. Just we should have a faith and look into detail as we are practicing since we start our life. So yes, being a thinkist together, I'm confident we will have a great journey and enjoy ourselves together. So let me start. Please have a close look to this short clip. And I request, try to think, speculate what this clip mean by. Okay, I hope you all might have a different perceptions, right? Like many of us observe beauty, wonder, plant, nature, growth, God's creation, etc., etc. You all are correct in one or another way. But we have many thoughts like why we are here, for what we are doing this, and most ultimate and one common question is what is life? Together, life is the aspects of growth, reproduction, activity, change, processes, acts, and reactions, adaptation, survival, mortality, and a lot more infinite. So, together, let's see all the events as a life. In my life, also, I used to have the same question: what is life? However, to my surprise, I really found the answer from my favorite, Scarlett Johansson. Not directly, as I'm a diehard fan of her and watch her movie Lucy many times just to have her. Literally, I don't remember how many times I have seen the movie for just her only. I'm watching her, but she answered. As you might see in the presentation by Morgan Freeman in the movie about the life, it means a lot. How? Let's see. I understood two terms there. And here, as you can see on the slide, the first, immortal, means living forever. It is the most not possible by anyone in any situation. Really not possible, at least to date. We or I can say no one doesn't know about the future, right? However, cancer cells can be classified in this category. And even using a fairness cream forever, it is never possible to have a fairness. The next option we have is reproduction. What is it? The action or process of copying something. The production of an offspring by sexual or asexual processes is mostly performed only in a favorable environment. And this is the only basis of diversity or evolution in life. And the ultimate option for any species is to immortal as my great great grandfather, great grandfather, grandfather, and my father is in me. Also in my child too. As humans, the most selfish species on this beautiful earth, we have developed a very favorable environment, and by adding or implementing the second phenomena, we are going to be more than 9 billion in probably next few years only. An event of reproduction leads to the transfer of hereditary material to the offspring to the child from a parent. We all know by what we all are very well know that the DNA, a magical paradoxical entity in the universe. Sometimes when a child has achieved the best, we used to say, "Mara jeevo chhe, mere jaisa hai, like me only." Likewise, many more things can be similar between parents and a child. That is called inheritance similarity, passing of a trait like a physic to other disorder. It can be your height, color, nature, to the disease condition like a diabetes, Alzheimer, blood pressure. This is about the similarity. But some differences are also remarkable. Contrary to inheritance, similarity, variation is a unique in a brood. We all know we are having some distinct feature that our parents don't have. These variations is the results of reordering in the DNA. If it is spelled correctly, we are so lucky. Even it is possible to get some good supremacy also. We speculated as in the X-Men and many sci-fi movie, but unfortunately, if it is misspelled, one has to suffer, pay it consequences. The term mutations shoots for both the conditions. However, misprinted mutations can accelerate some lethal genetic disorders. In crux, the science of heredity or genetics is the study of the two contradictory aspects of the nature. Yes, it plays an important role in the formation of a new species. As we think we are the only one and the super one. Here I must add, don't know why and how we are thinking like this. Pertaining to above context, coming to the topic also, I have composed this talk on DNA disharmony. I think you guys might guessed out. A genetic disorder. There is no doubt why I selected this topic. As said, Raja ne game te Rani. Just kidding, friends. It is really very near to my heart. As I started my research journey to work with this condition, really little known and lethal too, named Duchenne muscular dystrophy, abbreviated as DMD. We all know, and we all have an idea about the central dogma. Once the DNA, the Godfather ordered our little chefs, RNA, start to prepare food as a protein for our body and the functionality ultimately to the life. But what if an order gets jumbled? You can see in the slide that functionality will no longer exist like before. Leave us with the questions, why me? For this, we can set a term, genetic disorder. This is the highlight of this today's talk, the interactions. Let's start with the basic drawn a picture of this situation. In India, more than 50 million individuals are affected by genetic diseases. This number is going to mount up many times as the genetic predisposition in common diseases is more common toward our Indian community. In this genomic era, populace has restored their health and avoided unnecessary therapeutics with the help of a proper diagnosis. At present, in India, one of the biggest gain is the openness or availability of the molecular diagnostic tools and techniques for various genetic conditions. Gujarat also has very less work to view the burden of the same. Therefore, the information on which to develop strategies for understanding the genetic variants and the pathophysiological stipulations of this muscular dystrophy, especially Duchenne and Baker condition regarding the Gujarat population, especially in India, is again very essential for further management of the condition as well as to develop of new therapeutic approaches. By considering the above ground, we thought to undertake the study. So yes, what is muscular dystrophy? Yes, where I am interested. And the today's topic are a broad group of hereditary, progressive, degenerative disorders of a muscle which affect both human as well as animal. The word dystrophy is derived from the Greek word dys, means difficult or a faulty, and a trophy, which means nourish. Muscle weakness is a primary symptoms in all kind of MDs. Clinical and pathological criteria have been used in the past for classification, but nowadays MDs are mostly classified based on the genetic ground. In this genetic era, there are 9 types and more than 30 subtypes of muscular dystrophies are there. This disorder vary in the age of onset, severity and prototype of affected muscles. The cumulative evidence is suggested that an abnormal muscle surface membrane is the most likely pathology in all MDs, though this has not been proven yet. In humans, Duchenne muscular dystrophy and the less severe allelic form, that is Baker muscular dystrophy, are together termed as dystrophinopathies. So what is this dystrophinopathies, Duchenne and Baker muscular dystrophy? So yes, it is an X-linked recessive condition that affects 1 in 3,500 live male birth as a frequency. Affected individuals have mildly delayed motor milestones. Most are unable to run and jump properly due to proximal muscle weakness, which also results in the use of the classic Gover's maneuver. When arising from the floor, most patients are diagnosed at approximately 5 years of the age, when their physical abilities diverge markedly from their peers. Untreated muscle strength deteriorates and boy require to use of a wheelchair before their teens. Moreover, myelodic allelic form of this disease, that is Baker muscular dystrophy, as I mentioned earlier, which also causes ambulation, loss of ambulation at the age of 13 to 16 years. The pattern of inheritance can be quite useful in determining the mode of transmission for a given phenotype. Every genetist often studies the expression of a particular trait or phenotype in a family lineage, also known as a pedigree. In Duchenne and Baker muscular dystrophy, X-linked recessive inheritance is a mode of inheritance in which a mutation in a gene on the X chromosome causes the phenotype to be always expressed in male and in female who are homozygous for the gene mutation only, where females with one copy of the mutated gene are carriers. Hence, a positive family history, particularly in this dystrophinopathy, a maternal history is very useful and inexpensive tool to identify the patients as a high risk who may require a specific consideration for further management. As I have mentioned, discussed earlier, this is a genetic disorder. So which gene or genes are responsible for this? The answer is the longest human gene, DMD. The DMD gene, the meta gene of the human responsible for this dystrophinopathies, was the first isolated gene by the positional cloning method in 1986. The official name of this gene is dystrophin gene and the symbol is DMD gene located on XP21.2 on the X chromosome. DMD, the largest known human gene, is highly complex, covering 2.4 million base pairs with 79 exons containing at least 8 independent tissue-specific promoters and 2 poly-additional sites consisting of almost 0.8% of the total human genome or about 1.5% of the entire X chromosome. DMD gene, the largest known human gene, provides instruction for making a protein called dystrophin protein, and it is various isoform for specific tissues. Dystrophin, DMD protein, is found in muscle fiber. It is helical nature, allows it to like a spring or a shock absorbers. Dystrophin protein is located primarily in muscle, used for movement, mostly like in the skeletal muscle. Also, it also present in a smooth muscle and cardiac muscle too. Small amount of dystrophin are also present in nerve cells in the brain tissue. The protein dystrophin, measuring about 150 nanometer, is a rod-shaped cytoplasmic protein and is a vital part of the DGC that connects the cytoskeleton of a muscle fiber to the surrounding extracellular matrix through the sarcolemma. The dystrophin protein is the primary transcript of DMD metagene, measures about 2.4 million base pair, and it will take 16 hours to transcribe into a mature mRNA, measures about 14 kilobase pair, and codes a protein approx 427 kilodalton over comprises with 3684 amino acid residues. This sarcolemmal protein comprises only 0.002% of a total muscle protein, but up to 5% of a total membrane skeleton. Work as an anchoring, shock absorbing protein in muscle cell. Will make you more clear later in the talk with animations. Here, as I try to simplify with the linear structure of the dystrophin protein, emphasizing on its parts. Uh, to start this, element, there are some diagnostic strategy. We can say clinical symptoms are too important. Biochemical parameter can help us in screening the condition. Muscle biopsy, electrodiagnostic approaches, imaging tests, and yes, of course, a very latest and important, that is a molecular tools and techniques, the molecular test. Coming to the most interesting part of this talk, I'm sure you all will also appreciate it. It gives you glimpses of the condition, and we often times don't even look at them early. So let's begin by just watching a very brief animation movie in this 2 minutes. You saw a whole series of activities, symptoms, and life of the DMD patients that might you observe somewhere. I mean, when one child starts to fall down, what mental condition might parents have to face? When they know now the child age is not more than the 13 years, he will be wheelchair bound by the age of 12 to 13 years. We forget what this is just not a one child's, it is all about one family. The entire social structure might face the things. I actually want to show how much of the tough for the family to take care once a child. We do by labeling now, no more hope for such a condition because till date no such drugs are available for such condition. So you might question, then why one should withhold? Here I must put that nowadays with molecular knowledge, tools, fundamental, if we can diagnose the patients early in their early time, in their early age, we must have lesser severity to the life. We can, of course, increase life expectancy. They can live up to their fourth decade. Yes, and in this genetic era, we also having a few drugs, particular type of mutation in the market. And yes, of course, it is FDA approved too. Here we are dedicated working in this field and we are too much interested and we have very strong belief that we'll definitely reach this condition as soon as possible. I think now everyone's brain is swinging. Let me play a clip of this condition that together we have made for mass awareness and education purpose. Get pleasure of it.
A young gentleman and a beautiful lady were eternally bound by the holy ties as Saptapadini Mitsata. In the coming years, they were given the loved gift of parenthood and were blessed with a baby boy who was the center of the universe. And his tinkling laughter filled up their lives with unparalleled joy. Time flew by as the growing boy excelled in all areas of his life, and the family was happy with his achievements. One day, during a cricket match, the mother noticed that her boy started having difficulties keeping up with his friends. He seemed to walk much slower than his peers. This made his mother tensed and worried about his son. Soon the boy's parents noticed that their child had big calf muscles, had difficulty climbing the stairs, and had frequent falls while walking. The tears of joy were soon replaced by uncertainty and tension. There came a day when the young boy had to use his hands and arms as a support to stand and eventually lost his entire ability to move.
Hope you all have enjoyed it. And I'm sure you get the clear picture of this DMD condition and definitely helps us to kick off DMD in our society through some glimpses of research endeavors. So moving further, muscular dystrophy, as I mentioned, is a group of inherited disease that damage the muscle and muscle got weakened. Over and over at the time, this get degeneration and very weakness in the body. This damage and weakness are due to the lack of a protein called dystrophin, as I mentioned you earlier, which is necessary for the formal function of this muscles. The absence of this protein can cause a problem with walking and the muscle coordination. Let's know about the core and the actual problem with the DMD. The largest known human gene provides an instruction for making a protein called a dystrophin, primarily located in the muscle used for a movement and also found in heart as well as in the nerve cells in the brain and skeleton and cardiac muscle too. Dystrophin is a part of a group of a protein that work together to strengthen a muscle fiber to protect them from the injury by working as an anchoring protein and ultimately provide the strength to a muscle cell. If it is absence, we can see the condition like this. But if we get its altered mutation, absence due to defect in the DMD gene or a dystrophin gene, individual finally loses a muscle strength. Patients cannot live more than the 30 years of their life. Currently, there is no therapy or a drug exists for a DMD patients except for few palliative treatments as well as some are also in the conditional, we can say the conditional clinical approval. Yes, recently we also have few good hope for the patients, and we are sure soon will available to the mass also. During my research journey, I come to know some findings. As you can see in the slide, majority of the patients show deletion in the downstream region of the gene. The frequency of individual exon deletion is presented in the graph. Exon 50 was one of the most commonly observed mutation in this gene. Deletion started with exon 45, which was frequently as seen in our study. The highest deletion rate was the deletion 45 to 52. In the central part of the gene, the protein is studied DMD proband. The pattern of a mutation found in the Gujarat population is illustrated in this map, as you can see. We also observe one of the longest deletion, including exon 3 to 44. It is the longest and one of the unique deletion we have observed during the study. And of course, we have very controversial observation with the subject, that is, it shows a BMD kind of phenotype, though this much of big gene portion is missing. In our study cohort, out of the 20 family probands, 2 SIPs and a work proband of a known consanguineous marriages were not confirmed any deletion pattern with MPCR. In sum, the study was carried out in a limited number of Gujarati subjects. The method of MPCR is a doubtless tool for a diagnosis. Even though not rule out every mutation in a patient, thus we ought to use an alternative method like MLPA or high throughput next generation sequencing for diagnosis as well as for the carrier recognition. The location and the size of the deletion mutation found in the dystrophin gene do not indicate any correlation with the severity of the condition. Biochemical indices probably do not have any potential role in this diagnosis, even though they can be capable enough for screening and management of the condition. This study strongly emphasizes a need for further investigation into genotype-phenotype aspects of the Gujarati subject in order to provide a better diagnosis, prognostic, prenatal services, and counseling to the suffering family. So in this genomic era, we have a data on the human genome after successful completion of the Human Genome Project in the year 2003. The Human Genome Project gives us the capability, the ability for the first time to read nature's complete genetic blueprint on how human was made and evolved. The OMID, Online Mendelian Inheritance in MAIN, is a comprehensive, authoritative, and compendium of the human gene and genetic phenotypes that is freely available and updated frequently. The full text refer overviews in the OMID contain information on all known Mendelian disorders and over 15,000 genes. OMID focuses on the relationship between phenotype and genotype. In sum, we can say it is a guide and mentor in the genetics. So now, as I mentioned you earlier, DNA, a magical paradoxical entity on the universe. Why? Because DNA is a monomer for understanding genetics, or rather say life. So let's see the DNA. So DNA, as we all know, present inside every cell of our body. Each cell contains roughly 3 meters of the DNA. If unraveled all our DNA from all of the cells and laid out at the DNA end to end, the strand would stretch from the earth to the moon many times. The DNA is tightly coiled up, structured into 46 chromosomes. Our chromosomes are arranged in a pair that we have one from the mother and one from the father. There are approximately 3 into 10 raised to 9 base pair, that is a 3 billion chemical letters that known as bases, that are the A, T, C, G in the DNA, and that make unique code. If it is misspelled as I mentioned you earlier, that is a disease condition and sometimes as a evolutionary aspects. And they are everywhere in each and every cell of this body. This is a massive amount of information. It would fill 200 yellow pages in small type font. If you tried typing the whole genetic code out, it would take years, and yes, it would be without any breaks. But nowadays, having the latest tools and technology, we are ready to get the any human genome within a few days. DNA is made up of four building blocks. These are four letters, as you can see, A, T, C, G. Helps us to grow, develop, and functions. And what we are today ultimately. So once, as I mentioned, if it is altered, if it is get mutation, if it is get misspelled, then it is tough life. Nothing is important now. And this is the term genetic disorder. Now, might you have a question on how we can study a genetic disorder? I tell you, it is nowadays quite simple. To let me share my experience, it involves some steps such as profiling, pedigree analysis, genetic screening, etc. Before starting anything, it is very important to have empathy as well as faith and confidentiality. Propoma will help us to get a detailed information on the subject. Moving to the pedigree, as I mentioned, it is very basic, fundamental, and pocket-friendly practices to trace any genetic condition. We at our home even have practiced this. How? So most of you may be aware of Ambo, our family tree. Our parents used to see a family tree for marriages. And yes, they were so right. This is the concept of eugenics. How we'll talk later in this talk. And yes, it is so important. For further analysis, sample collection is must. Cytogenetic and molecular tools, techniques, and knowledge are used. And they are very beneficial for proper diagnosis of any condition. After diagnosis, still there is no hope for a better and the healthy life. Why? As I mentioned, there is no such drugs available. And again, the question arises the same. After all, why me? And what is the life and how to manage it? So here comes a very crucial element in this field. That is a genetic counseling. Genetic counseling is a responsible communication with the consultant probing. Genetic counseling is the process through which knowledge about the genetic aspects of the illness is shared by a trained professional with those who are at an increased risk or either having a heritable disorder or passing it on to their unborn offspring. A genetic counselor addresses the concern of the patients, their families, their healthcare providers, and supports the patients and their family dealing with this illness, providing them the knowledge, make them educating, and make them a decision maker. So as we can see in this slide, a proper discussion is a good option. I have put so many pictures uh, in this slide. Among them, many known personality you are aware about. And why I'm putting this, because they accomplish many things in their life and with challenging times and become a epitome or rather a role model for many of us. Here we are as a team to have a strength for such a personalities. As a human, different from all the species, we are curious and problem solving. Here the ultimate aim is to pass the message that is never, never give up. Because no one can beat the person who won't give up. Since last few years, working as an educator and a researcher, having a goal for the best life for all of us, I do believe in these three approaches together by heart. Because as it is the most fundamental approaches for the betterment of mankind. As you can see, euthanics, eugenics, and the euphemics. First, let's see what is euthanics. Euthanics which is the study of the improvement of a human functioning and well-being by an improvement of the environment, better nutrition, education, with suffer with sufficient amount of the medication. Moving to the next, that is the euphonics. What do you mean by euphonics? So, which is the study of the improvement of the human functioning. Sorry. Okay, coming to the euthanics, which is the study of the improvement of a human functioning and well-being by improvement of the environment, better nutrition, education, and with sufficient amount of medications. Next is very important, eugenics. Eugenics means improve the future generation by improving the germplasm of the existing population. In this, both heredity and a suitable healthy environment play an important role. As I mentioned earlier, as any species have such a condition, they try to be immortal as not possible directly. So one another option, that is the reproduction, as we discussed. In prior, hope you all are agreeing with me, play an important role in the development of the eugenics. The social movement claiming to improve the genetic feature of a human population through selective breeding, encouraging the marriages between the desirable candidate as a positive eugenics option. And on the other side, same time discouraging for an undesirable person as well as the sterilization, etc., option, they are a part of the negative eugenics. Based on the idea that it is possible to distinguish between superior and inferior elements of the society. Then, what is eugenics? The symptomatic treatment of the genetic diseases of a man. Example, you all know gene therapy, bone marrow transplant, molecular medicine, embryo therapy, etc. So I'm confident that you also now believe that at least once. Please think with these three concepts for a better world, let give the best world to the next generation. In sum, here I must add, and also you all have marked that all these three fundamentals have a prefix as eu: eugenics, euphonics, and euthanics. It means even. So it is definitely make a human life even and more fruitful one. Moving on the further, with above ground, we have a plan, or better say a hypothesis on which we are working for the betterment of the DMD condition all over the Gujarat, India, by putting with our full energy and blessings from the Almighty. So this is all about the approach to get rid of this, Duchenne muscular dystrophy. In addition to the aforementioned, for the welfare and the betterment of this, DMD patients, we have recently formed a team with the NGO, that is Indian Muscular Dystrophy Society, with more than 900 province subjects are involved as a family. We also try to alleviate the condition of the patients by providing supplement. They can avail the physiotherapy, wheelchair assistance, as well as their special need at the center. We regularly organize their supportives like a vaccination camps, awareness program, and cultural function to encouraging and to have their social interaction. At last, before goodbye, aujo, sayonara, the take-home message for all of us is please, take care of your DNA. It is an important, as you can see on the screen, in both the cases, once it is out of control, then there is no second chance. Even God cannot help us. So please take care. Here I would like to acknowledge team Gujarat University and HRDC for this wonderful opportunity, and of course to all my friends for listening to me. Further, we can have a lot more to know about from the various sources. These are the few good sources. So ultimately, in the conclusion, we started our lecture with the question, what is life? In a nutshell, I think you all now agree with me that life is a four-letter word that are the ATCG. And I hope I justified today's talk little, and by availing the Duchenne muscular dystrophy also in parallel, because after having and doing all the above, we used to have the same question from and most sufferers. Hope will soon have a good news for them, and hope you all have enjoyed it. Wishing you joy, happy, beautiful life ahead. I like to thank you all. So this is all about by my side. This is all about my research journey. Again, thank you all. Thank you so much, everybody. Stay safe, stay blessed. See you soon. Thank you all.